Now that we've learned more about DNA and DNA replication, we're going to learn about some of the ways we can manipulate DNA. In the next 3 videos, we're going to cover 6 different techniques, 5 that are commonly used in biotechnology, and 1 that is very new but very promising. This video will cover PCR and gel electrophoresis. The first technique we're going to talk about is the polymerase chain reaction, or PCR. The function of PCR is to copy many times a targeted stretch of DNA. PCR is essentially DNA replication, but in a test tube, and it targets a small portion of a chromosome. In order to perform PCR, we'll need several different components. The first component, the DNA that we're interested in copying. The second component, a DNA polymerase. For PCR, we use a special DNA polymerase called Taq polymerase. One of the steps in PCR heats the DNA up to high temperatures. That will denature most normal DNA polymerases. If we do that, every cycle we'll have to add fresh DNA polymerase. And most PCR runs go for 30 to 40 cycles. That would be very tedious. However, researchers were able to isolate the polymerase from a bacteria called Thermus aquaticus. That's what Taq stands for. Thermus aquaticus lives in boiling hot springs, and its DNA polymerase then has an optimal temperature that is very high, and it is not denatured by the high temperatures used in PCR. Because we use Taq polymerase, we can add the polymerase at the beginning of the reaction and let it run for 30 or 40 uninterrupted cycles. The third thing we'll need is nucleotides to build our new strands. And the fourth thing we'll need are 2 primers, one to target each DNA strand. Now let's look at how PCR works. The target sequence is the sequence we want to copy. The template DNA that we have is usually much longer than the region we're trying to copy. The DNA is heated up. This step is called denaturation, and that causes the 2 strands of DNA to separate. Once that's done, the primers that we've added to the mix will anneal, and once they have annealed, the third part is extension, where the DNA polymerase, again specifically Taq polymerase, will come in and build the next strand. Then we've gone from 1 to 2 copies of the DNA. Each time we do this, each round, we'll double the number of copies we have. After 2 rounds, we go from 2 to 4, and then 4 to 8, etc. Most PCR cycles have at least 30 rounds. And starting with 1, if I do 30 cycles of amplification, I will have 10 to the 9th copies. PCR is not usually the end of what people are trying to do. It's a step along the way. PCR in and of itself does not do an analysis of the DNA. However, it's a necessary pre-step because we're usually working with very small samples, maybe a drop of blood at a crime scene, or a small sample of tissue from an organism we're studying. PCR allows us to take that very small sample and create a very large sample that we can then work with. Another technique that is widely used is gel electrophoresis. In gel electrophoresis, DNA samples are put at the top of a gel, usually made of either agarose or polyacrylamide. An electrical current is then applied. Because DNA is negatively charged, it moves through the gel from the negative side to the positive side of the electrical current. Because the gel is porous, it's semi-solid. Think sort of like a very firm Jell-O jiggler. The DNA molecules can move through the gel, but larger molecules move more slowly and get stuck further towards the top of the well. Smaller molecules move further in the gel. That means this technique separates the DNA molecules by size. Any two bands of DNA that are at the same location in the gel are then the same size. This allows us to not only find out how large fragments of DNA are, but also to visualize our sample. Before the DNA is in the gel, it's just a clear sample of liquid. The technique of gel electrophoresis allows us to be able to see the DNA and check how large it is. Our take-home points for this video are that PCR allows scientists to make many copies of a targeted region of DNA. The process of PCR is similar to DNA replication, using nucleotides, primers, and a special DNA polymerase called Taq polymerase. Gel electrophoresis uses gel and an electrical current to separate DNA molecules by size. Gel electrophoresis is commonly used to visualize fragments of DNA and check their size.